Welcome to the deep dive. Today, we're really getting into the weeds on glycogen. It's the body's main way of storing carbohydrates, right, for later energy needs.
That's it. Sort of like how plants use starch. We use glycogen.
And we're aiming for a real mastery of this topic today. We want to cover everything from the ground up.
Absolutely. We'll unpack its structure, how it's built, its glycogenesis, how it's broken down glycogenolysis, the really complex ways it's all regulated.
And why it matters for health, right? Including those glycogen storage diseases.
Exactly. We've been digging into Harper's Illustrated Biochemistry, which gives a fantastic, detailed picture from molecules right up to the clinic.
Okay, so glycogen, it's not just everywhere in the body, is it? It's stored in specific places.
Primarily, yeah.
Yeah.
Liver and skeletal muscles hold the vast majority. There are smaller amounts in the brain too, mainly in astrocytes.
But there's a difference between liver and muscle stores, isn't there? Even though muscle holds more overall?
Right, that's a crucial point. The liver has a higher concentration of glycogen, like per gram of tissue. But because muscle tissue makes up so much more of our body weight,
Ah,
it actually stores the larger total amount. Think roughly 3/4 of our total glycogen. The numbers are something like 90 grams in the litter for an average person versus maybe 245 grams spread across all the muscles.
Okay, let's dig into that difference then. Why these two main storage sites? Why not just one big one?
Well, it comes down to their different jobs in keeping our blood sugar stable, what we call glucose homeostasis.
Right.
The liver glycogen, that's like the body's central glucose reserve. It's there to maintain blood glucose levels, especially between meals or when you're fasting overnight.
So the liver is sharing its glucose with the whole body.
Exactly.
Yeah.
Think about it, after an overnight fast, the liver's pumping out maybe 9 grams of glucose an hour. And about 2/3 of that, maybe 6 grams, is heading straight to the brain and red blood cells, tissues that absolutely rely on glucose.
So the liver's constantly adjusting, keeping things level.
Like a thermostat, yeah. A glucose thermostat for the blood.
But you mentioned about 90 grams total in the liver. That doesn't sound like a huge reserve if it's constantly being tapped.
It isn't, really. If that was our only source, we'd probably run out in, what, maybe 10 hours or so? That's why another process, gluconeogenesis, making new glucose from other things like amino acids or lactate, becomes so important, especially during longer fasts.
We'll get to that, I'm sure. But just focusing on liver glycogen for now, how much is left after, say, a normal night's sleep?
After a typical 6 to 8 hour fast, maybe down to around 20 grams.
Yeah.
And if you go longer, like over 18 hours, it's pretty much depleted. Really shows the impact of fasting.
Okay, so liver. Central sharing depot. What about the muscles then? They've got way more total glycogen, but they're not sharing it.
Nope, they're not. Muscle glycogen is strictly for local use. It's a fuel source for that muscle cell.
Ah, okay.
When muscle breaks down glycogen, it makes glucose 1-phosphate, which jumps right into glycolysis to make ATP for muscle contraction.
So why can't it release glucose like the liver does?
Because muscle cells lack a key enzyme. Glucose 6-phosphatase, that's the enzyme the liver uses to snip off the phosphate group, allowing free glucose to leave the cell. Muscles just don't have it.
Got it. So muscle glycogen is like a private fuel tank for that specific muscle.
A high-performance fuel tank, yeah. Ready for immediate use during activity.
Okay, that makes sense. Liver for the system, muscle for itself. You mentioned glycolysis in muscle produces pyruvate. What happens to that, especially during intense exercise? The source mentioned lactate and alanine.
Ah, yes, the Cori cycle. It's a really neat bit of metabolic cooperation. During intense work, muscle glycolysis cranks out pyruvate. Some of that gets converted to alanine, some gets reduced to lactate.
And these leave the muscle.
They do. They travel through the blood to the liver. And the liver can take that lactate and alanine and use them as building blocks for gluconeogenesis to make new glucose.
Which it can then release back into the blood.
Exactly. It's like recycling energy potential between muscle and liver. A very efficient system.
A metabolic shuttle service. So after we eat, how do we refill these stores, especially the livers? Is it just soaking up glucose from the meal?
Well, yes, but it's actually a bit more nuanced than that. Our source suggests only about half of liver glycogen replenishment after a meal comes directly from glucose taken up from the blood.
Only half. So where does the rest come from?
It comes indirectly from those gluconeogenic precursors we just talked about, lactate, alanine, other glucogenic amino acids. The liver converts those into glucose, or glucose precursors first, and then incorporates them into glycogen.
Huh. So it's not just a one-way street of glucose in, glycogen out. The liver's also using these other building blocks.
Precisely. It's called indirect glycogen synthesis, a key concept if you really want to master this.
That is fascinating. Changes how you picture post-meal metabolism. What about the brain's glycogen? You mentioned small stores there.
Yeah, mostly in astrocytes, there's support cells for neurons. Their exact role is still being, fully worked out. But the idea is they provide a local energy buffer, a quick glucose supply for neurons if blood glucose dips slightly or demand spikes locally.
And could problems with brain glycogen explain some symptoms in those glycogen storage diseases?
It's thought to be a contributing factor, yes.
Mm-hmm.
If astrocytes can't manage their glycogen properly, it could impact neuronal function, leading to some of the neurological issues seen in certain GSDs.
It really sounds like an incredibly sensitive, balanced system. Let's talk about deliberately messing with that balance, like athletes doing carbohydrate loading.
Right, that's a classic example of manipulating glycogen stores. The goal is to maximize muscle glycogen.
How does it work exactly?
Typically, you start with intense exercise to really deplete the muscle glycogen stores. Then, for a few days, you eat a very high carbohydrate diet while tapering down your training. This combination really pushes the body to synthesize glycogen, often leading to levels higher than normal supercompensation, they call it. Interestingly, the glycogen formed might even be less branched.
Fewer branches. Why?
The rapid synthesis might outpace the branching enzyme activity somewhat, but the main goal is just maximizing the total amount stored, giving more fuel for endurance events. But athletes need to get the timing right, or it can backfire.
Supercharging the muscle tanks. What about eating carbs during exercise? The source said that helps endurance too.
It does, primarily by helping maintain blood glucose levels. If you're supplying glucose from outside,
Like from a sports drink or gel?
exactly, then the liver doesn't have to break down its own glycogen quite as fast to keep blood sugar up. And by keeping blood glucose stable, you also reduce the rate at which the muscles burn through their glycogen stores, especially those fast-twitch fibers.
So you spare the muscle's limited reserves.
Right, you can keep going longer.
Makes sense. One last point on the basics before we dive into synthesis. Water. Glycogen holds a lot of water, apparently.
It really does. But about 3 grams of water gets bound up for every 1 gram of glycogen stored.
3 grams. That's significant.
It is, and it explains why people often see rapid weight loss initially when they start a very low-carb diet. As glycogen stores get used up, that bound water is released.
So that first few pounds isn't necessarily fat loss.
Often, a good chunk of it is just water weight coming off along with the glycogen. Good to keep in mind.
Okay, foundational understanding established. Let's get into the machinery. How do we actually build glycogen, glycogenesis? Mostly in liver and muscle, you said.
Correct. And step 1 is getting glucose into the cell and trapping it there. That means phosphorylating it, adding a phosphate group to make glucose 6-phosphate.
And the enzyme doing that is different in muscle versus liver.
Slightly, yeah. Muscle uses hexokinase. The liver uses a related enzyme called glucokinase. Both do the same job initially. Stick a phosphate on glucose, that traps it inside.
Trapped inside, got it. What's next for this glucose 6-phosphate?
It needs a slight rearrangement. The phosphate group has to move from the number 6 carbon to the number 1 carbon. This isomerization is done by an enzyme called phosphoglucometase.
Just shifting the phosphate over.
Pretty much. It actually involves a little temporary intermediate glucose 1,6-bisphosphate, and the enzyme itself gets transiently phosphorylated. But the end result is glucose 1-phosphate.
Okay, glucose 1-phosphate. Now what? How does it get ready to be added to the chain?
This is the activation step. Glucose 1-phosphate reacts with UTP, that's uridine triphosphate, similar to ATP.
Ah, okay.
This reaction forms UDP glucose, or UDP-GLC, plus pyrophosphate. The enzyme is UDP-GLC pyrophosphorylase. Think of UDP glucose as the activated glucose donor. It's carrying the glucose unit in a high-energy linkage, ready for transfer.
UDP glucose, our building block. And pyrophosphate is also produced? The source said this step is irreversible.
Effectively irreversible, yes, because that pyrophosphate produced is immediately split into 2 inorganic phosphates by another enzyme, pyrophosphatase.
Ah, removing a product pulls the reaction forward.
Exactly, Le Chatelier's principle in action. It ensures the pathway flows strongly towards making UDP glucose. And interestingly, the UDP-GLC pyrophosphorylase enzyme itself isn't considered a major control point. It's usually abundant and works efficiently.
So we have our activated glucose unit, UDP glucose. How do we actually start a new glycogen molecule? We can't just stick UDP glucose molecules together, can we?
No, you need a primer. And that's where a fascinating protein called glycogen then comes in.
Glycogen.
Yeah. It's a protein that acts as the seed for every new glycogen molecule. It actually catalyzes its own glucosylation. It attaches the first glucose from UDP glucose onto one of its own tyrosine amino acids.
Wow, it starts itself.
It does. And then it adds about 7 more glucose units, also from UDP glucose, linking them with those straight alpha-1 arrow bonds. This short glucose chain attached to glycogen is the primer.
The foundation.
Exactly. And glycogen stays right there, buried in the core of the finished glycogen granule. There's even a specific glycogen storage disease, GSD-XV, linked to defects in glycogenin, mainly affecting the heart. Shows how critical that first step is.
Incredible. A self-starting protein core. Okay, so once the glycogen and primer is ready, what takes over?
Now the main workhorse, glycogen synthase, steps in. It takes UDP glucose and adds the glucose unit to the non-reducing end of the growing chain, forming another alpha-1 or 4 bond and releasing UDP.
So it just keeps adding glucose units, making the straight chains longer.
That's its job, adding glucose after glucose via those 1 or 4 links, extending the branches initiated by glycogen.
But glycogen isn't just straight chains, it's highly branched. Where do the branches come from?
That requires a different enzyme. The branching enzyme, technically called amylo 134 transglycopylase.
Itchy name.
Yeah. So once glycogen synthase has built a chain that's at least, say, 11 glucose units long, the branching enzyme comes along. It breaks an alpha-1 or bond within that chain, takes a segment of at least 6 glucose units, and transfers it to attach via an alpha-1 or 6 bond to a glucose residue on the same or a nearby chain.
Creating a branch point.
Exactly. It creates that alpha-1 or 6 linkage. Now you have a new non-reducing end where glycogen synthase can start adding more glucose units via 1 to 1 or 4 links, and eventually that new branch can also be branched. That's how you get that complex tree-like structure.
And those branches are crucial, right? For quick access.
Absolutely vital.
Yeah.
Having all those branches means many, many non-reducing ends. Glycogen synthase can add glucose to all of them simultaneously during synthesis, and crucially, the breakdown enzymes can remove glucose from all of them simultaneously during glycogenolysis. It massively speeds things up.
Makes sense. Efficiency through structure. Okay, we've built our glycogen. Now, how do we break it down when we need the glucose? Glycogenolysis. And you stress this isn't just glycogenesis in reverse.
Definitely not. It's a separate pathway with its own key enzymes. The main player, the rate-limiting enzyme for breakdown, is glycogen phosphorylase.
Phosphorylase, what does it do?
It attacks those alpha-1 linkages at the non-reducing ends. But instead of using water like in digestion, it uses inorganic phosphate, a process called phosphorylases.
Ah, phosphorylasis.
Right. So it breaks the bond and adds a phosphate at the same time, releasing glucose 1-phosphate. Not free glucose, but glucose 1-phosphate.
And there are different versions of this enzyme.
Yes, isoenzymes in liver, muscle, and brain, encoded by different genes, reflects their slightly different regulatory needs in each tissue. It also requires a coenzyme, pyridoxal phosphate, derived from vitamin B6.
Vitamin B6. Interesting.
Yeah, and unusually, it's the phosphate group on the pyridoxal phosphate that's directly involved in the catalytic mechanism here.
Okay, so phosphorylase chews away at the 1,4 bonds, releasing glucose 1-phosphate. What happens when it hits a branch point, one of those 1E6 links? It can't break that, right?
Correct. Phosphorylase stops about 4 glucose units away from an alpha-1 or 6 branch. That's where the debranching enzyme comes in.
The unbrancher.
Pretty much. It actually has 2 different enzyme activities on the same protein molecule. First, it acts as a transferase. It takes a little block of 3 glucose units from the branch and transfers it to the end of another nearby chain, extending that chain's 1 or 4 linkage.
Okay. It moves most of the branch over.
Right. That leaves just a single glucose residue attached by the alpha-1 or 6 bond. Then, the second activity of the debranching enzyme, an alpha-1 or 6 glycosidase activity, kicks in. It hydrolyzes that 1 or 6 bond, releasing that single glucose unit as free glucose.
So we do get some free glucose directly from breakdown, just from the branch points.
A small amount, yes. Maybe about 8–10% of the total glucose released. The vast majority comes off as glucose 1-phosphate via phosphorylase.
And once the branch is removed, phosphorylase can continue down the now linear chain.
Exactly. The debranching enzyme clears the way for phosphorylase to keep working until it hits the next branch point.
Okay, so we're getting mostly glucose 1-phosphate. What happens to that?
It gets converted back to glucose 6-phosphate by that same enzyme we saw in synthesis, phosphoglucometase, just running in the reverse direction now.
Back to glucose 6-phosphate. And this is where liver and muscle part ways again, metabolically speaking.
Precisely. This is the crucial junction. In the liver, there's that enzyme we mentioned earlier,
Okay.
glucose 6-phosphatase.
The one muscle doesn't have.
Right. Glucose 6-phosphatase removes the phosphate group from glucose 6-phosphate, producing free glucose. This free glucose can then be transported out of the liver cell into the bloodstream to raise blood glucose levels. Interestingly, this enzyme is located in the endoplasmic reticulum membrane, not the cytosol.
Okay.
Defects in either the enzyme itself or the transporter that gets glucose 6-phosphate into the ER cause types of type I glycogen storage disease.
And in muscle, no glucose 6-phosphatase.
So the glucose 6-phosphate produced from glycogen breakdown in muscle cannot be released as free glucose. It stays in the muscle cell and enters glycolysis to generate ATP for muscle activity.
Fueling the muscle directly. Got it. What about that other breakdown route mentioned, lysosomes?
Yes. Besides the main cyclic pathway, lysosomes can engulf glycogen granules. Inside the lysosome, an enzyme called acid maltase or lysosomal alpha-glucosidase can break down glycogen completely to glucose. It can handle both alpha-1 OR and alpha-1,6 bonds.
Is this a major pathway?
Probably not for moment-to-moment energy needs, but it might be important, maybe especially in newborns. A deficiency in this enzyme causes Pompe disease, which is type 2 GSD, where glycogen builds up in lysosomes, causing severe problems, particularly in muscle and heart tissue. Importantly, this lysosomal breakdown isn't under the same tight hormonal control as the cytosolic pathway.
Okay, that clarifies the different breakdown routes. Now, the really complex part, regulation. How is all this synthesis and breakdown controlled so precisely? The source highlights cyclic AMP, CMP.
CAMP is absolutely central. It's a second messenger molecule that acts as a key integrator, coordinating glycogenolysis and glycogenesis. The core principle is reciprocal regulation, meaning the control mechanisms work in opposite directions on the key enzymes. Generally, phosphorylation adding a phosphate group activates glycogen phosphorylase breakdown, but inactivates glycogen synthesis.
And dephosphorylation does the reverse.
Exactly. Dephosphorylation inactivates phosphorylase and activates synthase. It's like a molecular switch, ensuring you're not simultaneously trying hard to build and break down glycogen, which would be incredibly wasteful, like pressing the accelerator and the brake at the same time.
Makes sense. And CAMP levels themselves are controlled by hormones.
Right. Hormones like epinephrine, norepinephrine, and importantly in the liver, glucagon, bind to cell surface receptors. This activates an enzyme called adenyl cyclase, which converts ATP into CAMP. So hormone signal leads to increased CAMP inside the cell.
But the signal isn't permanent?
No, because CAMP is constantly being broken down back to AMP by enzymes called phosphodiesterases. Insulin, for instance, can increase the activity of phosphodiesterase in the liver, which lowers CAMP levels and shifts the balance towards glycogen synthesis.
Okay, let's focus on regulating phosphorylase first, the breakdown enzyme. Liver versus muscle regulation differs.
It does, reflecting their different jobs. Liver phosphorylase needs to respond primarily to blood glucose levels the body's overall need. Muscle phosphorylase is more attuned to the immediate energy state within that muscle cell.
But both are controlled by phosphorylation.
Yes, both exist in a less active B form, dephosphorylated, and a more active A form, phosphorylated. The enzyme phosphorylase kinase adds the phosphate to activate it, and protein phosphatase 1 removes it to deactivate it.
Is it just on-off, or are there other controls? Allosteric regulation?
Oh yes, absolutely. Allosteric control provides another layer of fine-tuning. In both liver and muscle, high levels of ATP and glucose 6-phosphate signs of high energy inhibit the active phosphorylase A. Makes sense, right? If you have plenty of energy, slow down the fuel release.
Yeah, feedback inhibition.
Exactly. The liver has an additional layer. Free glucose itself can also inhibit liver phosphorylase A. So if blood glucose is high, the liver automatically slows down its own glucose release.
Clever. What about muscle?
Muscle phosphorylase B, the normally less active form, gets strongly activated by AMP. High AMP levels are a sign of low energy charge in the muscle. ATP has been used up, producing ADP, which gets converted to AMP. So even before hormonal signals fully kick in to phosphorylate it, if the muscle's energy drops, AMP can directly boost glycogen breakdown.
So muscle responds very quickly to its own energy needs via AMP.
Very quickly. It highlights the different priorities. Liver managing systemic glucose, muscle managing its own immediate fuel supply.
Okay, back to CAMP. How does it actually trigger the phosphorylation of phosphorylase? You mentioned phosphorylase kinase.
Right, it's a cascade. Increased CAMP activates another enzyme called CAMP-dependent protein kinase, or PKA.
Protein kinase A.
That's the one. PKA then phosphorylates and activates phosphorylase kinase. Then, the now active phosphorylase kinase phosphorylates glycogen phosphorylase B, converting it to the active phosphorylase A.
A cascade, like amplification.
Exactly. Each step amplifies the signal. A few hormone molecules can lead to the activation of many phosphorylase molecules very quickly. It's a way to get a big response from a small initial trigger.
And the triggers are different hormones for liver and muscle?
Correct. In the liver, the main trigger for raising KMP is glucagon, released when blood sugar is low. Muscle doesn't respond to glucagon.
Muscle responds to
Epinephrine, or adrenaline. Released during stress, exercise, fight, or flight situations. It tells the muscle to get ready for action by mobilizing its fuel stores via KMP.
So different signals, same internal CAMP-MP pathway tailored to tissue function, but you also mentioned calcium in muscle, linking contraction and breakdown.
Yes, this is incredibly elegant. The rise in calcium ions inside the muscle cell that triggers contraction also helps activate glycogen breakdown.
How does that work?
Remember phosphorylase kinase, the enzyme that activates phosphorylase? It's a complex protein. One of its subunits, the delta subunit, is actually calmodulin.
The calcium binding protein.
The very same. When calcium floods the cytosol during contraction, it binds to this calmodulin subunit on phosphorylase kinase. This binding partially activates the kinase, even if it hasn't been phosphorylated by pKa yet.
Wow, so contraction itself gives glycogen breakdown a kickstart?
Exactly. It ensures fuel supply is immediately coupled to fuel demand during exercise. Full activation needs both calcium and phosphorylation, but calcium provides that instant link.
Amazing integration. What about the liver? Any non-CAMP ways to trigger breakdown there?
There are. For instance, norepinephrine acting on alpha-1 adrenergic receptors can increase calcium inside liver cells, which then activates phosphorylase kinase via calmodulin, similar to muscle, though this pathway might be less prominent in humans than in some other animals.
Okay.
Also, hormones like vasopressin and angiotensin acetic can stimulate liver glycogenolysis through calcium-dependent pathways or other signaling routes like the PIP2 pathway, so there are backup systems.
Always good to have backups. Now, how do we turn all this off? Protein phosphatase 1 or PP1 seems key.
PP1 is the major phosphatase involved here. It removes the phosphates from both phosphorylase inactivating it and phosphorylase kinase inactivating it, thus shutting down the breakdown cascade.
But its activity is regulated too.
Yes. PP1 itself can be inhibited by another protein called inhibitor 1. Inhibitor 1 only works after it's been phosphorylated by, you guessed it, pKa, the CAMP-dependent kinase.
So high CAMP not only turns on breakdown, but also helps turn off the enzyme that stops breakdown.
Exactly. It reinforces the signal. Insulin works partly by promoting the activity of PP1, helping to shut down glycogenolysis. Plus, insulin increases glucose uptake, raising glucose 6-phosphate, which allosterically inhibits phosphorylase. Multiple levels of control.
It's dizzying.
Yeah.
Okay, let's switch to the other side. Glycogen synthase, the builder enzyme. Its regulation is the mirror image of phosphorylase, right?
Pretty much, yes. Again, different isoforms exist in liver, muscle, brain. And like phosphorylase, it's controlled by phosphorylation, but the effects are opposite.
So phosphorylation inactivates synthase.
Correct. The dephosphorylated form, synthase A, is the active form. The phosphorylated form, synthase B, is generally inactive.
And it's not just pKa doing the phosphorylating.
No, glycogen synthase is a target for a whole bunch of kinases, at least 6 are mentioned, phosphorylating it on multiple different sites, at least 9 serine residues. This includes pKa, but also calcium calmodulin-dependent kinases, including phosphorylase kinase itself.
Wow, so phosphorylase kinase activates breakdown and helps inactivate synthesis.
Yes, contributing to that reciprocal control. The fact that pKa phosphorylates and inactivates synthase directly links high CAMP levels to shutting down glycogen synthesis while simultaneously turning on breakdown.
And insulin promotes synthesis?
Yes, insulin promotes the dephosphorylation of synthase back to its active A form, mainly by activating protein phosphatase 1. High glucose 6-phosphate levels resulting from insulin-stimulated glucose uptake also help activate synthase phosphatase activity.
So PP1 is the hero for synthesis, removing those inhibitory phosphates from synthase.
PP1 is crucial for activating synthase, just as it's crucial for inactivating phosphorylase and phosphorylase kinase. It coordinates the switch between breakdown and synthesis.
Let's try to summarize this balance then. High CMP means pKa is active.
Which means phosphorylase kinase is active, phosphorylase is active, so breakdown is ON, and glycogen synthase is inactive, so synthesis is O off F. Plus, PP1 is inhibited via inhibitor 1.
Okay, and low KMP.
pKa is less active, PP1 becomes more active. PP1 then dephosphorylates phosphorylase kinase, phosphorylase, and glycogen synthase. So breakdown turns OF and synthesis turns on.
Beautifully coordinated. Insulin pushes things towards synthesis by lowering CAMP and boosting PP1.
Exactly. It's a synchronous control system. And that multi-site phosphorylation adds layers of fine-tuning, modulating how sensitive the enzymes are to these primary on-off signals.
And high glucose itself helps amplify insulin's effect, especially in the liver.
Yes, high glucose entering the liver leads to more glucose 6-phosphate via glucokinase. This G6P inhibits breakdown, phosphorylase, and helps activate synthase, reinforcing insulin's actions.
The liver's rapid response seems so critical, especially thinking about exercise starting. Preventing hypoglycemia sounds paramount.
Absolutely vital. Severe hypoglycemia is dangerous. The liver needs to ramp up glucose output instantly when demand increases. Glucogenesis helps too, but it's slower.
Right.
During moderate exercise, the balance between liver glucose output from glycogenolysis and gluconeogenesis and muscle glucose uptake is remarkably well maintained, usually with little change in blood glucose. This is thanks to those hormonal shifts. Less insulin, more glucagon, and the muscle's ability to take up glucose even without high insulin when it's contracting.
An incredible metabolic balancing act. Let's shift to the clinical side now. Hypoglycemia and diabetes seems like a major practical issue related to all this.
It really is. A key goal in diabetes management is keeping blood glucose near normal to avoid long-term complications. But the treatments themselves, especially insulin, carry the risk of causing hypoglycemia, low blood sugar.
And sometimes people stop recognizing the warning signs.
Yes, that's hypoglycemia unawareness. Very dangerous. People might not feel the usual sweating, tremors, or confusion until their blood sugar is dangerously low. We distinguish between non-severe hypo, where you feel symptoms and can treat yourself.
Like eating something sugary.
Right. And severe hypo, where function is impaired, maybe confusion, seizures, needing help from others.
And glucagon injections are used then.
Yes. Glucagon is a rescue treatment for severe hypo. It strongly stimulates liver glycogenolysis, rapidly raising blood glucose. It can be life-saving.
Okay, then there are the glycogen storage diseases, GSDs, inherited problems with glycogen itself.
Yes, a whole group of genetic disorders where there's either too much glycogen, structurally abnormal glycogen, or an inability to break it down properly because a specific enzyme is deficient.
The source had a big table listing many types.
It did. There are quite a few labeled with Roman numerals, type I, von Gierke, type 2, Pomp, type 3, Cori, type V, McArdle, and so on. Each results from a defect in a different enzyme involved in glycogen metabolism.
And they affect different tissues.
Yes, depending on which enzyme is faulty and where it's most important. Some primarily affect the liver, causing enlargement and hypoglycemia, like type 1. Others mainly affect muscle, causing weakness and exercise intolerance, like type V McArdle disease. Type 2, Pomp, affects lysosomes everywhere, causing widespread issues, especially in heart and muscle.
They sound quite varied.
They are very heterogeneous. Most are autosomal recessive, needing 2 copies of the bad gene, though a few are X-linked, while individually rare, recognizing them is important.
How are they diagnosed and managed?
Diagnosis often involves clinical suspicion based on symptoms, maybe enzyme tests on tissue biopsies, and increasingly, genetic testing. Newborn screening is catching some earlier now. Management often relies heavily on nutrition things like frequent meals, sometimes cornstarch overnight, specific diets depending on the type to prevent hypoglycemia and manage symptoms. Early diagnosis and proper management can make a huge difference in preventing long-term problems.
It really highlights how crucial these pathways are when you see the consequences of them going wrong.
Absolutely. It underscores the importance of understanding this biochemistry for clinical practice. For anyone wanting to dig even deeper, the references in our source material are excellent starting points.
This has been an incredibly detailed exploration. Glycogen metabolism is clearly far more intricate and elegantly regulated than just simple sugar storage.
It truly is. It's fundamental to energy balance, glucose homeostasis, fueling everything from brain function to muscle contraction. We've seen the different roles of liver and muscle stores, the complex web of hormonal and allosteric control.
Phosphorylation cascades, the phosphatases.
Right. And the serious clinical implications when things go awry, like in GSDs or with hypoglycemia and diabetes.
It definitely leaves you thinking, if glycogen regulation is this complex, how many other metabolic pathways have this level of intricate control and crosstalk happening constantly to keep everything running smoothly?
That's a great question. It really speaks to the incredible interconnectedness of our biochemistry, doesn't it? Constantly adapting, constantly balancing.
A fascinating deep dive. Thanks for joining us today.